Right, I'm quite pleased to see this decent number of people there, actually. I think this campus has been so quiet, I'm surprised to see that many people. Delighted to have Jess Crawshaw here from Oxford. So Jess did a PhD in Melbourne and then moved straight to Oxford, and very happily has just secured a position at the Francis Crick in London. So she's going to be talking to us about the eye.
Thanks, Danny. So I'm going to be talking about pharmacokinetic, pharmacodynamic models of a particular treatment for a disease called macular degeneration. And I'm going to slowly lead us into this by talking first about what the eye is, what the disease is, and how this drug works. So this is a lovely picture of the eye. In the center, we've got the macula here at the back of our retina. The macular is where we've got the most photoreceptors concentrated. So this is the center of our vision, the most refined area. The rest of the retina has photoreceptors, but not as many. So this is our peripheral vision, it's more fuzzy. So if anything goes wrong with this macula here, it's going to mess with what is the most important part of the eye. So age-related macular degeneration is one of the diseases that can mess with this area. Basically, this is a nice healthy eye with an OCT image. Then we've got dry AMD, where we've got this kind of fatty protein stuff building up at the back of the eye near the retina. This stuff's called drusen, and it's bad. Then the disease progresses to wet AMD, where we start getting the blood vessels involved, and it starts releasing fluid all around the retina, causing mass disruption. What this looks like to a patient can vary. So here's a couple of examples. Things can go a bit fuzzy and blurred because you've got this disorientation of your photoreceptors, and you can have death of the photoreceptors resulting in just the blank spots in your vision. And as the name suggests, this is something that happens to older people. So it's not fun at all. To understand this disease, we have to understand a little bit about what the retinal looks like. We've got this nice structured retina here. We've got the choroid, a bunch of blood vessels down there. We've got the retinal pigment and epithelium, which seems, unobtrusive, but that they are the bad cells that start this cascade here. And then we've got our photoreceptors. The RPE here, the retinal pigment epithelium, are the supporting cells for the photoreceptors and keep things pumping along nicely through life. So these RPE cells start to get stressed and tired, and they release this fatty lipid byproduct called drusen. It gets built up here, and we see that it starts to cause a bit of a disorientation of the photoreceptors. Things are bad here. This is dry macular degeneration, but it can cause a block between the blood supply and the rest of the retina. This results in neovascularisation, which these new blood vessels, they're immature, they're leaky, they're leaking stuff all over the place. So we've got an accumulation of liquid where we shouldn't have it. And this, completely disrupts our photoreceptors and it kills them. So this is really bad. Our treatment strategies for this are primarily injections into the eye, straight into the vitreous. So we call these intravitreal injections. And we inject a anti-VEGF drug. So we've got all this neovascularisation because of an abundance of VEGF. We inject this drug, hopefully it soaks up all the VEGF running around and removes it so we can have things going from bad to less bad to okay. Notably, we've still got the drusen here. We can't fix that, it's there. The best way to get rid of that is laser ablation of regions of the retina, and there's no coming back. You can't get your photoreceptors back from that. You've got a fixed number of photoreceptors for life. Everybody has the same number of photoreceptors, except for some select members of the population who have one more, which is really cool. So this seems great. We just do an injection into the eye and it fixes the problem, but temporarily. The fluid dynamics within the eye means that we've got rapid elimination through the front of the eye and into the back of the eye. And what this is going to look like in our graphs from experimental data is we've got a rapid drop of VEGF. This is VEGF concentration in the aqueous. The aqueous is this little bit at the front of the eye. That's much easier to sample from rather than the vitreous. If you take stuff from the vitreous, you destroy the eye. So we sample from the aqueous and we see that we've got a rapid drop in VEGF. This is followed by a hasty rebound because the drug is eliminated from the eye, the VEGF can come back and do what it wants. This results in frequent injections between every 1 to 3 months. As such, there is an extensive interest in improving this drug. So we need to talk a little bit about this drug development pipeline if we want to think about improving drugs. We've obviously got the basic modeling and in vitro studies in the dish. Everything is okay there. Then we go to safety experiments in small animals like rats and rabbits, but their eyes aren't like human eyes. Their VEGF is also different. We then upgrade to the monkey eye to see if it works. Obviously, there's lots of money going at this process. Things can get rejected at any stage. We test in the non-human primates, primarily cynoculus monkeys. And then from there, if that's all good, we go to the human trial. The human trial is the most expensive stage, obviously, and it's difficult to get ethics approval, et cetera, et cetera. So this is the transition I'm focused on between monkey to human studies. So the main question of my work is, can we use the monkey models and experiments to predict and understand the human data? To some degree, yeah, we can, but can we use the models to add more power to this prediction and extend this prediction? So going back to what the problem is, too many needles in the eye, not enough duration of effect. Let's start talking about how this drug actually works. The drug that we're focusing on, called ranibizumab, so we can understand what is not good enough about this drug and what aspects we need to improve. So ranibizumab is one of the many drugs you can use to treat this disease. It is the most expensive, and it is the one that my my industry collaborators use, so we focus on that one. But if you've got AMD, I might recommend using rolosivimab because it's several orders of magnitude less expensive. It lasts slightly less time as well, unfortunately. Anyway, so this drug, you inject it into the eye and it binds to the VEGF we've got running around here. It can bind once or it can bind twice. So we've got mass action kinetics going on here. So we've got the equations for that. We've got partially bound VEGF and double bound. This can happen in the vitreous, which is the center of the eye, or the aqueous, which is in the front of the eye. And we've got elimination from the vitreous to the aqueous of each of these species. There is VEGF, ranibizumab, partially bound and double bound. And we've got outflow. So there's all of our parameters in our model. There's our model. We convert it into a set of ODEs and voila, there we have it. We've got 8 nonlinear ODEs. Here are all our mass action terms. Here are all of our elimination rates, and here are all of the parameters running around in the model. If we are able to make these parameters better, we can make the drug better. So we've got to understand what these parameters are. Also, this is what it looks like. If we just pick a bunch of random parameters, we see that, as expected, we've got our VEGF going down and then rebounding. This is the ranibizumab in the aqueous. It exponentially decreases, as you would expect when you've got elimination of something. So that's a log axis here. So to understand these parameters, we've got 3 steps, as one should do when they're going to do Bayesian inference, which is the final step here. So testing structural identifiability, sensitivity analysis, and then finally the Bayesian inference. If anybody is ever going to go into Bayesian inference, always remember to do these 2 at the start because it will save you a lot of time and pain at the other end. So structural identifiability. I've got this slide in more as I get asked about this every time I give this talk, so I thought I would briefly talk about it. We use Castro and DeBoer's method, which basically utilizes the invariance of these parameters under scaling transformations. And we find in the end we have structural identifiability after going through a whole bunch of pages of algebra, and that's nice. Moving on to the next part, structural sensitivity analysis. We use eFast, which is extremely handy and not too hard to use. Basically, it looks at the variance in the parameters and then how they come out in the model to basically tell you which parameters at the other end are the most important. So I looked at a couple of different aspects of our ODEs here to figure out which parameters are most important. So firstly, I looked at this rebound time in our aqueous VEGF curve, and I looked at the point of inflection here. So they're two important points clinically people care about for how long this drug is in their eye for. And we found that the elimination of the drug is the most important parameter telling us how long the drug is in the eye. So that makes sense. It feels a little bit stupidly easy, but it tells us something really important. So hidden here, we've got a little equation saying that the elimination rate of this drug is proportional to the molecular weight. Experimentally, they've shown that the molecular weight is the most important factor when it comes to drug retention. So this is saying our model agrees with the experimental data, which I find a really good sign that our model is quite accurate, or it's going quite well, it's along the right lines. So there we go, there's our elimination rate. I then looked at this from another approach. I considered the importance of each individual parameter over time. And again, we see the elimination rate, which is this red curve along here, is always the most important. So when we look at our sensitivity analysis, we get first order and total sensitivities. Total sensitivities basically means the sensitivity of one parameter varying with other parameters. So the first order is just one parameter varying. The total order is the parameter varying with other parameters varying. And it shows us that the elimination of ranibizumab is always the most important regardless. So with all this information, knowing that I have structural identifiability and which parameter I should get the most information out, we go to our Bayesian inference. I've been told that this is a more mathematical route, not really statistical. That's fine. I don't usually talk too much about how Bayesian inference works, but the key points is it does work, which is great. This is Bayes' rule. It's basically multiplying the evidence we have, experimental data, by our prior information. And we've got some idea of what these parameters should be from experimental literature. And if we numerically just do this again and again and again, we form some kind of posterior. So this log likelihood is a set of parameters, how likely they are given the information we have. And we've got some information. We multiply them again, 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 and we get this nice red posterior here. I use something called PINES, probabilistic inference on noisy time series. It's a software package that's being developed by the ComSci group at Oxford. It's very useful, it's very user-friendly, we recommend. So these are all of the ingredients in my Bayesian inference. I've got my data here. This is monkey data. We've got our ranibizumab concentration in the vitreous and our VEGF concentration in the vitreous. So of all of the species, all of the combinations, we've got 8 ODEs. So 4 species in 2 compartments, and we've only got data for 2 of them. Put that together with our model and selecting our parameters gives us this equation here, which is the log likelihood. And I guess the takeaway from this here is it's a normal distribution. Then we've got our priors. These values are being pulled from the literature. And from that, I put together a table of what I think should be sufficiently wide priors. Priors literally means your prior information or knowledge about the system. So that's what I've got. In the end, we've basically got a normal times by some other distribution that we've got. So these are our results. We've got our posterior distributions coming out at the end. So these were what I originally thought the values would be, pulling them out of the literature. And then we've got the actual values that they are here. So there's a couple of things that I want to pull your attention to here. Firstly, there is our elimination rate. It's kind of different to what we initially expected, 0.1 compared to 0.28. The fits I have are excellent and I'm quite confident about them. So these values here, they've got little stars on them because they're kind of guesses based on a loose model that we have, like a loose relationship between some other experimental values. So I'm far more confident about what I've got on the other end here. The other two things that we need to discuss is this KD. So if you look at what KD is from the literature, the range is stupid, right? There is several orders of magnitude different. The value I get is kind of in the middle. It's 1,495. I know that value very well because I've spent a lot of time trying to see whether I can get it to be the same as any of these experimental values. And every time it goes back to this same value, no matter where I start my search from. So I'm very happy about that. The next thing we need to talk about is K-off. This value is quite rubbish. It's saying that it's distributed around 0. I should say K-off is the dissociation rate between ranibizumab and VEGF. Saying it's 0 doesn't make sense. It doesn't make sense in the model. It doesn't make sense anywhere. So if we then go and look at how the fits are, that's quite good. So we've got I've got here our mean plus our credence interval from our results. For our the plots where we've got data, it's a really narrow credence interval, which says, you know, we're very confident of those data sets, of the results in those areas. For each of the others, it's a little more wide. However, given we've got no data, it's doing pretty good. So back to our controversial KD. I want to know why our value is so different to all of the other values. So I've gone and I've rerun my Bayesian inference, except I've set KD to be a given value. And that value has gone from 0 to 4,000, just to see how each of the other parameters would change. And basically we see they seem to be pretty reasonable at the value we think it is. At the small values, where most of our literature says it is, our values of other parameters are ridiculous, and our log likelihood is also really negative. The larger your log likelihood is, the better. It basically means your result is closer to the data. So we see it increases until it starts to cut off here when the increase is pretty minimal. So if we look at different KD values plotted with the data, you can see that for really small values of KD, the results are rubbish. For larger values of KD, the results are better. There are several reasons why the experimental and the modeling KD are so different. So I should say KD is a ratio between the association and dissociation of these molecules. So it's temperature dependent, and none of the experimental studies capture the correct temperature and pH of the eye. So this KD has an important effect on how quickly the drug is eliminated, and most of the assumptions of how this is working are based on in vitro studies that aren't accurate. I should say in vitro means in a petri dish, so not in realistic conditions. So we've talked about a whole bunch of monkey stuff. What about human data? So here is our human data. We've taken this from Saunders et al. From 2016, and we've got lots of data. It looks all over the place, and it is. This data isn't the worst, but most certainly isn't the best. We've also only got VEGF in the aqueous. So we've got about 31 participants, which is a good number. And we can see if we draw a curve in PowerPoint, it looks like it could fit. It has similar behavior to the monkey data, so that's nice. We've got to upgrade our techniques. Previously, we were just using Bayesian inference. Now we need to upgrade to a hierarchical approach, meaning we've got the population level and we've got the individual level. Without describing too deeply what hierarchical Bayesian inference is, I just need to say we've got a whole sweep of parameters. Before we had, I think, 8. Now we've got 8 times 32 plus 8. Plus 1. So we've got more parameters, and that's going to scale up with the more participants you have. In a subsequent study I'm doing, I've got 300 participants, and it blows up really quickly. Anyway, we've got parameters on the population level. We've got a mean parameter and our standard deviation parameter, so how that varies across the population. So for clearance, we've got these are our priors here, I should say. We've got some assumption of what the mean of that parameter is across the population, and that's our prior there. And we've got some assumption of how that parameter varies across the population as well. We've got the same for all of our other parameters. These ones here, I'm assuming they don't vary across the population because these two parameters are dependent on the drug, not the individual. Then we've got our individual parameters. So how these parameters, they're set for each individual and they are directly inferred based on the data. The population level parameters are inferred based on the individual level parameters. So it's getting a lot of information, which is getting their information from their individual participants' data. I'm also going to make the comment that age-related macular degeneration doesn't happen in small children like I've got shown here, but it felt a little bit cruel to use a cartoon old man. So we run the hierarchical Bayesian inference and we get these results. And this side looks amazing. This side looks tragic, specifically these two. So this is our KD, and it seems to say it's around 0. Which makes no sense. And then we've got our payoff. It's the same as the prior. So we've got no information from that. So whilst I've shown you that I have practical identifiability, clearly from this data set, I've got structural identifiability. From this data set, I don't have practical identifiability. So there's not much I can do to get these parameters. I could change data sets, and that's what I've done in subsequent studies. But for now, I need to just set these values to be the same as the monkey. Our industry collaborators say they should be the same, that should not vary between monkey and human. Great, so I've gone and set those 2 values and happily moved on with my life. In doing so, I've got far better results to the extent I have sat down and properly plotted them out. So along the x-axis in each of these plots, I have the mean value of the population, and along the y-axis, I've got the standard deviation. So these are our hyperparameters, and you've kind of got to think about this for a second, what this looks like. But if you don't want to, the best way to think about it is you just take the peak value, and that will give you your mean and standard deviation of how a given parameter varies across the population. But this is the information we can get for the data, and I'm pretty happy with that. We then go and look at how the parameters vary for each individual. And for most, they're pretty good. These here are the elimination rates for VEGF, partially bound, and fully bound. And they're really consistent across the population, which honestly is surprising. We've then got our VEGF input, which is the rate that your retina is producing VEGF. And we do see quite a lot of variation here. And that's mathematically going to be a really fun problem, because in humans, they've got the disease. In the monkey data, there is no disease. They're just healthy monkeys. In the humans, this value is going to vary between human, and you can see that. But it's also going to vary across time between injections. And we haven't accounted for that in this data, and to do so is going to be very, very difficult, and we don't have enough data. So I'm excited to tackle that problem later. And then we've got clearance rates, which vary a little bit, but no one really cares. The elimination of ranibizumab is the key parameter here. We've got a lot of variation across the population. within, from a clinical approach, they see that there is a lot of variation in each patient of how long this drug lasts. And that's why I said at the start, you need an injection every 1 to 3 months. That's a huge range, right? And this is the reason here. Basically, the smaller your elimination rate of ranibizumab is, the longer it's going to last, the longer the drug half-life is, which makes sense, right? We think that the reason why we've got so much variation in this parameter is because there is a lot of covariates here. So age is the main one. In a future study, I'm going to see if I can correlate the variation in this particular variable with all the other factors that are going on. So the next thing I want to draw our attention to in this slide is up here, I've got our monkey parameters. So these values here are taken straight from the inference with the monkey. You can see there's quite a difference for the elimination rates. Our VEGF input for the monkey, which is here, is nice and narrow and kind of consistent with our human data. You can see it's a lot more narrow. That's because they're healthy monkeys. Makes sense. There's no variation. And our clearance rate, it should be different because they're different species, different eyes. What we really want to do is understand how the monkey elimination rates, which are these parameters over here, relate to the human elimination rates, these parameters over here. If we can find a value which converts one to the other, then we can take this monkey data, all this monkey fitting, scale the parameters we get at the end, and then predict what happens in the human. And I have done that, which is amazing. Here are the values down here. So if we take these values up here and scale them by a factor that's proportional to the ratio of the vitreous volume squared, so the volume of the center of the eye, you can get these values down here. I should have put the equation up there. I apologize for not doing it, but basically there's this simple scaling factor you can use to get the values to be approximately the same, which is really exciting for future work. Here are our fits for a selection of the humans, and you can see, I think they're pretty good. So in some humans, you get very few data points, like in this one here. In other humans, you get a whole bunch of data points, but it doesn't seem to matter too much. You can always get the fit. There is also quite a variation in the disease state. So some humans have a lot going on, a lot of VEGF in there, like this one here. So its VEGF concentration is about 2, whereas other humans have it closer to 1.2 over here. Oh, here we go, there's a 0.75. So despite the variation in these humans, this approach captures everything. So it's really good. Yep, so there's the scaling issue, and yeah, we get the same general response between humans and monkeys. So that seems to answer everything we need. We've got the correct general behavior described by the data, and we had that before we started anything. But now we can go, well, we get these results from the monkey, we can transfer to the human to predict the human more strongly than before with these scaling factors. And these scaling factors are due to the differences in the size of the eye. Geometric difference and fluid flow differences are the two parameters altering things there. So then the next steps from here is a bigger and better data set. So the next data set we're using is the Archway trial, and we've got over 300 patients in that data set. We've also got ranibizumab data. This is really exciting because, you know, more data is good, but it's also a pain in the neck because 300 patients makes things go very, very slowly. So unfortunately, currently confidential, and I can't really talk about it other than to show you blurred plots that don't actually have the right data underneath them, or else I'd get in trouble. All right, so basically the conclusions from all this work show that the drug size has the greatest impact. We can infer the parameter distributions in a simple 2-compartmental model that I showed you. Our values of KD are very different to the in vitro ones, and I'm pretty sure these ones are more correct. Unfortunately, the pharmacodynamic parameters are not are currently attainable through the human data. So that's our KD and our K-off. And the elimination rates are the most notable difference between the humans and the monkeys. I'd like to acknowledge my supervisors and my collaborators at La Hoffmann Roche in Switzerland. And thank you so much for attending my talk. And I welcome any and all questions.
Thank you very much, Jess.
Thanks, Danny.
Any questions?
Hi, that was a very good talk. I was just wondering, you said that the monkeys are all healthy.
Yeah.
The data.
Yep.
How does that data match to people that are actually ill?